This video is about how to estimate size and scale in cell images. Example 5. This is an image of phage, viruses, bacterial viruses, attacking a bacterium. The image is from Wikipedia. The question is, what's the width of this image from one end to the other of the double-headed blue arrow? And pause if you want to work this for yourself. Here, there are two things from the bacterial ruler: viral capsids and a bacterium. On the bacterial ruler, a capsid is about 100 nanometers, and a bacterium is about 1 micron in diameter. The diameter of the bacterium is about the width of the image, so the width of the image should be about 1 micron. Can we check our work? This time, you can. The capsids are about 100 nanometers, and if I measure carefully, it would take about 14 capsids to cross the image. I screenshot some capsids and then inserted them in a row next to each other and labeled them 1, 2, 3, all the way up to 14. Doing it this way, about 14 times 100 nanometers equals 1400 nanometers, or 1.4 microns. That suggests that the whole image is 1.4 microns instead of 1. But most phage capsids are a little smaller than 100 nanometers, and so 1 micron is probably closer to correct. It doesn't matter. Both methods give about the same answer, and that's what we want. They don't need to be identical. We're estimating. Here's a mystery image. What's the diameter of these circles? I've hidden the scale bar in the lower right of the image. It's covered in a white rectangle. If you don't know what it is, then you have to start making guesses. You need to start trying to identify something in the image, and ideally it would be something on the bacterial ruler. So make 3 guesses if you can. Decide which guess is most likely, which is next most likely, and which is least likely. And pause here to make your guesses and write them down. Did you pause? Did you make your guesses? If you didn't, pause and guess at least one thing. These would be my guesses. First, that the black lines are membranes, lipid bilayers or plasma membrane. Circles and continuous line in electron micrograph images are very often lipid membranes. 2, the circles could be mitochondria. Some of them look a bit like they might be a cut through a mitochondria, but mitochondria usually are not all circular, and all of these are. In addition, one of them is incomplete. If you look at the one in the middle, it's actually connected to the long line that goes all the way across. That would be surprising for a mitochondrion. So it doesn't really seem like it's a mitochondrion. It's possible the circles could be cells or bacteria, but there's enough resolution that I would expect to be able to see at least some unevenness inside these, and all of them look either an even gray or an even pale. I don't really see any structures. So that makes me think that it's unlikely that they're cells or bacteria. Okay, so let's take the first one. If the lines are lipid membranes, then the lines are about 10 nanometers thick, or 7 to 8 nanometers if you remember the detail on the bacterial ruler. Membranes are a little bit thinner than 10 nanometers. So step 3, there's space for about 4 thicknesses of the black lines inside each circle. I put 4 arrows here, or about the distance apart of those black lines, to show you that you'd fit about 4 of them in there. The interior of this circle is 4 times 10 nanometers, about 40 nanometers. For the diameter of the circle, you have to add 10 nanometers for the membrane on one side, say the left, and 10 nanometers from the membrane on the other side, say the right. 10 plus 40 plus 10 equals 60. The total is about 60 nanometers. Or, if we used 7 to 8 nanometers for the membranes, the total would be about 50 nanometers. How close are we? Now I've uncovered the scale bar on the lower right, 200 nanometers, that long black bar. The circles look to me to be about 1/4 as wide as the scale bar, and I could believe that some of them might be close to 1/3 as wide as the scale bar. I don't think most of them are much less than that. If they're 1/4 of 200 nanometers, that equals 50 nanometers. If they're 1/3 of 200 nanometers, that equals 67 nanometers. We estimated somewhere around 50 or 60. We were very close. What you're actually looking at in the mystery image I gave you here was vesicles of neurotransmitter at nerves. These are the chemicals that are being released from one nerve cell to another. The one that looks like it's open, that's attached to the middle line, that's one that has just released or is releasing its contents across the synaptic cleft to the next neuron. I hope that you can go on and do your own bacterial ruler problems. If you go on Google Images and search on electron micrograph animal cell or plant cell or virus, or on photomicrograph, you'll find images at many different scales, all the way from sections through organs with many, many cells in them, down to sections of tiny parts of cells like this one, where you should be able to find these images and start estimating sizes for yourself. And every time you're reading a textbook or noticing something where you have an image, I hope you start looking at it and thinking about size and scale. You'll quickly notice that in line drawings of things like proteins interacting with ions or proteins near cells, that a lot of the structures that are shown are not to scale. If they show a protein next to a cell, it should be a nearly invisible dot if you can see a whole cell. If they show an ion next to that, it would be invisible. And yet, when we show line drawings, we often illustrate them as something much bigger than they really would be, because if we didn't, you'd never understand. It isn't a bad thing that we often don't show things to scale when we show line drawings, but it's important for you to recognize and not be fooled. I hope that if you keep practicing these, you'll wind up discovering that, like my students and like me, whenever you think of biological structures, you'll start realizing very easily what their sizes and size relationships are.